about coffee plant nutrition and soil fertility in Hawaii. So some of you might know, but our coffee yields have been declining for the last couple of decades. We've seen a pretty substantial decline of about 50%. Back in 2000, we were at about 1,200 kg of green coffee per hectare. And in 2024, we're down to about 650 kg per hectare. So it's a pretty substantial decline. And this is in contrast to a lot of the other large coffee growing regions around the world. You can see on this figure here that Asia and South America are increasing their coffee production, and Central America and Africa are at least stabilized. But we are continuing to decline year over year. So there are likely a lot of reasons for these declines. As we all know, we've been hit really hard by coffee berry borer and coffee leaf rust, as well as coffee root knot nematode. Those are probably the big three that are contributing to declines of coffee production here in Hawaii. We also have some pretty old orchards, at least in the Kona side, and throughout the rest of the state. And then, of course, we have weather, whether that be, really dry years or really wet years. So likely a lot of different contributions to these declining yields, but it's a big issue for us. Here in Hawaii, most of the research that's been done on coffee has been primarily focused on pests and diseases. As you can see from this pie chart, there's been 121 studies done so far on Hawaiian coffee. Most of those on coffee pests and diseases, also on just general agronomy, beneficial insects. But you can see a very small percentage of that research has been focused on plant nutrition and coffee soil fertility. So the big questions for this study, we wanted to know if poor plant nutrition and low soil fertility are limiting Hawaii's coffee yields and by how much. And we wanted to know if we are actually fertilizing Hawaii coffee correctly or if there are some improvements that we can make. So we did the first state line baseline of coffee nutrition in Hawaii. We focused on seven different growing regions across the state, Kauai, Oahu, West Maui, East Maui, and then here on the Big Island, we looked at Hilo, Kona, and Kau. So the red in the graph are just the different coffee areas. And then each one of those circles is a sampling site for us. So you can see that the larger farms like Kauai Coffee, we sampled really throughout the entire estate so that we could get a good representation across all the different elevations and the different varieties that they might have planted there. And then in the smaller coffee growing regions like Oahu, which primarily we just sampled from Dole, and then some of the smaller regions like Hilo, we might have only sampled one farm. But we did try to sample across all the major regions, and this was a one-time sampling that we did. We ended up with 236 soil samples and 139 leaf samples. The reason for the differences in those sample numbers, most farms historically have only been doing soil sampling, and this leaf tissue sampling is really sort of a newer thing for Hawaii. And so for the farms that we were able to get some of their data records from recent years, they might have only sampled soil. And so that's why we had a larger number of soil samples versus the leaf samples. And then we did a second part to this study where we focused on Hawaii Island, and we sampled a total of 21 farms, and we sampled those farms across 3 different years, so 21, 22, and 23. So that we got 3 consecutive years of data, both for soil and leaf tissue. And then we also collected data on the fertilizers that they were applying and also their coffee yields. We followed the CTAR recommended sampling methods for soil and leaf tissue, and we conducted the sampling for the farms that we did sample and where we didn't just collect their recent records. We did the sampling from sometime between December and March in the post-harvest. For the coffee leaves, we sampled from the 3rd pair of fully emerged leaves, sort of at the mid-level. And then for the soil, we sampled halfway between the trunk and the drip line, and we dug down to about 6 inches deep and collected soil from that region. And then if it was a small farm, we combined those 10 to 15 trees into a composite sample. If it was a large farm, we might have collected many composites, like I said, from like Kauai coffee, where we sampled from a lot of different plots. So each one of those plots might have been 10 to 15 trees, and that was combined into a single composite. But then we did multiple composites throughout the entire area. So starting to get into some results, probably not too surprising, but not all Hawaiian soils behave the same. And this has a lot to do with the geography of the islands. We're in a hotspot archipelago. So as you move from from Hawaii Island to the northwest, the islands get older and older. So you can see in this figure, the different colors represent the different soil types. So you can see on Hawaii Island and on East Maui, the soil, there's only a couple different main soil types, andisols and histosols, and then the gray are the lava flows. So as you increase in island age, you also increase in soil diversity. So as you get to West Maui and Oahu and Kauai, you can see all those different colors. They each represent different soil types. So a lot more different kinds of soil as the lava flows age. They go from these large soil particles, the basalt particles over time, they become more and more clay rich, and those particles get smaller and smaller. So on the young soils, you sort of get these sandy loam textures that drain really fast. So you get a lot of leaching, a lot of nutrient leaching. And on the young islands, you tend to have low phosphorus, but you have high nitrogen. And then as you move up the island change, you get more and more different soil types. They become more and more clay rich. So that's that really red soil that you might remember seeing on Kauai. That's mostly clay. And it has better water holding capacity. And they tend to be more enriched in metals. So like iron oxides and aluminum, they tend to have lower pH, and they also tend to have lower organic matter. So all of these things matter when we're talking about soil and plant nutrition, because these factors really affect how the plants are able to uptake different nutrients and what's available to them. So now getting into some soil nutrient deficiency results. What nutrients are most deficient in our Hawaii coffee soils? So this figure, on the x-axis, you have all the different soil nutrients. And then on the y-axis, we have soil nutrient status. So anything down here in this bottom rung is a low available nutrient status, and then we have adequate and high. And then each one of these dots represents a single plot that was sampled. Kauai is purple, Oahu is red, Maui is green, Hilo's yellow, and then Kona is in black and Kau is gray. So they're all color coded. So you can see that Kauai is really deficient in nitrogen, and also Oahu and some of the Maui sites. And then Kona, Kau, we tend to be more deficient in like calcium and phosphorus and magnesium, and also potassium. If we look across the whole state, nitrogen was deficient in 85% of all the samples that we looked at. Calcium was deficient in 67%, phosphorus in 61%, magnesium in 48%, and potassium in 22% of all samples. So again, as I pointed out in the previous slide, Kauai, Oahu, and Maui, they tend to be nitrogen limited, whereas young Hawaii Island tends to be more phosphorus, calcium, magnesium, and potassium limited. And then looking at leaf nutrient deficiencies. So we see some different patterns. Out of all the samples that we looked at, potassium was the most deficient. In almost all the samples we looked at, potassium was deficient. And then phosphorus in 65%, zinc in 59%, copper in 42%, and nitrogen in 38%. So potassium is the single most widespread deficiency in all of Hawaii coffee. So different from the soil results, which is pretty important. Then we also wanted to know if the soil nutrients predict the plant nutrition. So if you remember, we sort of saw two different sides to the soil nutrition and plant nutrition sides of the story when we look at the soil versus when we look at the leaves. So we wanted to know if the soil and the leaves, for any nutrients, if they told the same story. We did find that soil testing worked well for nitrogen, magnesium, zinc, and sulfur. So a high correlation between the soil result and the test results for those 4 different nutrients. Then as we start to look at the other nutrients, we see two different stories being told. The soil's telling us one thing and the leaf tissue is telling us something different. So soil testing is really a poor predictor for these other major nutrients: potassium, phosphorus, calcium, iron, copper, manganese, and boron. So the main story here is that if you're relying on soil tests alone, you may be missing a lot of different deficiencies that could be present in your plants that your soils just aren't telling you about. And then if you remember, we also focused on Hawaii Island for the second part of our study, where we sampled from 21 different farms over 3 consecutive years, because we wanted to know if the soil and leaf nutrients differed from year to year, or if any of them stayed the same. What we found, which you can see on this graph, is that in the soil, we saw a lot of fluctuation in potassium, calcium, and phosphorus from year to year, whereas things like pH, carbon, nitrogen, zinc, magnesium, those all tended to stay the same from year to year. And then when we looked in the leaf tissue, we found that a lot of the micronutrients differed a lot. So things like zinc, magnesium. Sorry, let me minimize this real quick here. And then things like the leaf macronutrients, so nitrogen, phosphorus, and potassium tended to be more stable from year to year. So again, one-time soil tests are likely unreliable for things like phosphorus and potassium, and leaf testing is going to give you a more stable signal for these major nutrients. We also wanted to know what growers were applying. So again, this was the data from Hawaii Island. Probably not too surprising, but most of the fertilizer applications were NPK. So you can see that about 60% of all fertilizer applications were nitrogen, about 45% were potassium, and about 30% were phosphorus. Very few applications of calcium, magnesium, sulfur, and copper. And then all of the micronutrients that were being applied were primarily through Cafe DAC, which is foliar blend. And so that's why the boron, manganese, iron, zinc, and molybdenum were all in about the same amounts. And then when we looked at those NPK fertilizers, we also found that most of those fertilizers were like a 1-to-1-to-1 or a 1-to-0-0, so sort of like similar amounts of NPK, and very few of the applications tended to have higher ratios of phosphorus or potassium. So basically what we found is that there's really no correlation between the nutrient levels in the plant and the amount and different types of fertilizer that people are applying. So this is just a super basic table that shows some of the most common deficiencies on the left-hand side, and then some of the most commonly applied fertilizers. So potassium is the most commonly deficient nutrient in our coffee plants, but people tend to be applying mostly nitrogen. So they might be overspending on nitrogen, but underspending on potassium, which most of our coffee farms really need here, and they're just not getting enough of. So a pretty big mismatch between deficiencies and what's being applied. And more fertilizer doesn't always necessarily mean that you are going to get better nutrition, because you may not be looking at your test results or even doing your testing to be able to inform you what you should be applying. So it's not always about how much you apply, it's more so about what you're applying and are you targeting the right deficiencies on your farm. So what should growers be doing? Leaf and soil testing should be routine and they should be done at least once per year. I think that leaf testing actually should probably be done more than once a year, maybe once in the post-harvest and then maybe once in the summertime before you get into the major harvest. Just so you can sort of get an idea of what you're deficient in, when the harvest is done and then right before you go into harvest again. But at a minimum, if you could do those at least once per year, I think that would be great. Prioritizing potassium because it really is the most widespread deficiency in plants throughout the entire state. And then more smaller and more frequent applications so that we reduce nutrient loss through leaching, especially here on young Hawaii Island where we have, pretty high rainfall and really large fragment sizes. A lot of those nutrients are just moving through the soils and not necessarily being absorbed. It's also important to not just apply your nutrients blindly, that you're actually using these test results to guide what you're applying and adjusting your fertilizer strategy to your soil type and your rainfall. So again, here on Hawaii Island, where we have a lot of leaching, you might want to focus on smaller applications more frequently of NPK, and then also monitoring your calcium and magnesium ratios. And then on older soils, you should be watching for N limitation and thinking more about adding organic matter, monitoring your pH, which might be really low, and looking at your metal content as well. So that is all I have for you today. Big thanks to the Specialty Crop Research Initiative grant, which provided the funding for this project, and all of the great field support that I had, and also to Luis for providing a lot of soil and tissue samples for this project as well. So thank you all, and I'm happy to answer any questions.